We completed post-gem RT Dynabead cleanup. Now we will perform cDNA amplification.
Amp mix comes in two sizes. We have amp mix, which we vortex, flick the reagents down, and place on ice.
Next, we will select the primers to use for the amplification.
If we are only preparing a gene expression library, we select the cDNA primers from the Chromium NextGem single-cell 3' HT Gem Kit v3.1. If we are preparing gene expression and feature barcode libraries, we will select one of the feature cDNA primers from the single-cell 3' feature barcode kit. Whether you select feature cDNA primers 1, 2, or 3 depends on the application of interest.
Today, we are performing a gene expression experiment without feature barcode technology.
Therefore, we will use the cDNA primers from the Chromium NextGem single-cell 3' HT Gem Kit v3.1. We select the cDNA primers, equilibrate to room temperature, vortex, briefly spin down, and return to room temperature.
Now we will prepare the cDNA amplification reaction mix on ice.
We will add the reagents in the order shown in the user guide. Calculate the volume of each reagent that you will need for your number of samples, including 10% reagent volume overage. First, we will add the amp mix. Then add the cDNA primers we previously selected. Vortex thoroughly, and briefly spin down. Add 65 microliters of cDNA amplification reaction mix to each tube containing 35 microliters of purified Gem RT product. Pipette mix 15 times with the pipette set to 90 microliters and briefly spin down. Load the tube strips into a thermal cycler that can accommodate at least 100 microliters reaction volume and proceed with the following incubation protocol. The optimal cycle number is a trade-off between generating sufficient final mass for libraries and minimizing PCR amplification artifacts. Select PCR cycles based on the target recovery per gem well and not on the estimated number of cells in the two tubes containing the split sample. The cDNA cycles should be reduced if sampling large numbers of cells. Remove the cDNA amplification product from the thermal cycler. Store at 4 degrees Celsius for up to 72 hours, or minus 20 degrees Celsius for up to 1 week, or proceed to the next step.
We completed cDNA amplification. Next, we will perform SPRIselect cleanup.